Hi students, today we will start the our next session, that is antihistaminic agents. In that part 4, that is a structure active relationship of the antihistaminic agent. Okay, we will for this session we will see the H1 antagonist SAR activity, that is structure activity relationship of the H1 antagonist. Okay, this is very important for exam point of view. So first of all, we'll see the structure, exactly what is the structure is there. So this is the basic structure here given. So according to this structure, there are 4 different subparts included in this structure. Okay, first of all, I will clear that the structures. This is the aryl group, one part is there. Okay, that is we can substitute here any phenyl ring or any other heterocyclic rings also we can substitute. Okay, according to that. Next is a portion of the X, that is we can attach any elements like oxygen, carbon atoms, then nitrogen, etc. Okay, this is the first part, this is the second part. Okay, this is the first and this is the second part. Next is the ethylene bridge, it's the third part, subpart, that is the ethylene bridge. Okay, we can here increasing the chain ethylene bridge. Okay, so it will be increasing activity or decreasing activity, we will see in this video. Okay, first of all, clear that how many parts are there of structures. So the third one is the ethylene bridge and the last part is the amino group. Okay, so amino group, it should be a tertiary amino groups should be there for the increasing activity of H1 antagonist. Okay, so clear, it is clear now. There are 4 parts of there in a basic structure. First of all, aryl group, second is X, that is the different types of elements. Next is the ethylene bridge and last is the amino group. So we can add here, suppose first of all we will discuss about this aryl groups. We can add any aryl groups like phenyl rings or heterocyclic compounds here. Okay, that is the, and mostly the diaryl, this is the, and 2 means diaryl groups definitely it needs to effective activity for the H1 antagonist. Okay, without that it will not showing the proper activity. Okay, so diaryl group substitution is essential for activity, clear that. And the example is the diphenhydramine, simple structure is there, diphenhydramine, it is a one type of examples. Next is the substitution of the, suppose we substituted here, suppose one phenyl groups here. Okay, in that we attach the another halogen atoms like chlorine, bromine, etc. So it also increasing the activity. Okay, it will be increasing the activity. Example is bromodiphenhydramine. Okay, we can add that, it will increase the activity. So this is the substitution here, first part. Next is the X substitutions, different elements are there, oxygen, carbon, nitrogen. Other than these oxygen, carbon and nitrogen atoms, if suppose we attach other elements, then definitely it will be decreasing the activity, okay, of H1 antagonist. Clear? Next is the ethylene bridge. If suppose we increasing the chain of ethylene bridge, increasing the branching of the chain, this is the branching, suppose this is the branching. Okay, increasing on the carbon atoms here. If suppose we increase this, then definitely there is a decreasing activity. Only the exception case is promethazine. Okay, promethazine is a exception case. For that purpose, it will not impact on the activity of the H1 antagonist in the case of promethazine. Okay. Next, the last part is the amino group, and the amino group, the tertiary amino group is essential for the our H1 antagonist activity. Okay, for this structure. So basically we will necessary of this tertiary amino group for the essential activity. So this is the structural activity relationship about the H1 antagonist. Simple way is there, just you have understand the 4 different subparts, that is the one aryl group, second is the different types of elements, third is the ethylene bridge, and last is the amino group. Okay, these are the important points. Just keep in mind and study well. Okay. Thank you so much. Those who are not subscribed this channel, please subscribe this channel for further education videos. Okay. Thank you so much.